Bismillahirrahmanirrahim. We start discussion on nerve and muscle. Nerve and muscle. Nervous tissue is composed of neurons and neuroglia. So nervous system is composed of neurons and neuroglia. First, I discuss about the neuroglia or glial tissue. Neuroglia consists of two types of cells, microglia and macroglia. So microglia and macroglia are two types of the glial tissue. Microglia resemble macrophages. So microglia resembles macrophages. But keep in mind, microglia are not derived from macrophages. So it is not derived from the macrophages. Microglia has a phagocytic function. The phagocytic function of the microglia, that is, that they take up, they engulf, they remove the cell debris. If cell is damaged, its debris is removed by the microglia having phagocytic function. So this was about the microglia. The other types of the other type of cells is macroglia, and there are three types of macroglia: astrocytes, Schwann cells, and oligodendrogliocytes. So astrocytes, Schwann cells, and oligodendrogliocytes, these are the types of the macroglia. Astrocytes, they take part in the formation of blood-brain barrier. So astrocytes take part in the formation of the blood-brain barrier. So what is blood-brain barrier? The brain capillaries have selective permeability. Brain capillaries have selective permeability. These are permeable to small molecules and lipid-soluble substance. So brain capillaries are permeable to small molecules and lipid-soluble substances. But brain capillaries are not permeable to large molecules, protein, dyes, many antibiotics, right? So this selective impermeability, or as we say selective permeability of the brain capillaries, is the blood-brain barrier. Many antibiotics, many drugs, they can't cross the blood-brain barrier. Dopamine, when given by mouth, absorbed into the blood, it can't cross the blood-brain barrier. So this selective impermeability of the brain capillaries is the basis of the blood-brain barrier. So this is about the function of the astrocytes. Schwann cells, they make myelin sheath in the peripheral nervous system. So along the peripheral nerves, myelin sheath is formed by the Schwann cells. The third type of macroglia is oligodendrogliocytes. Oligodendrogliocytes. gliocytes. These take part in the formation of myelin sheath in the central nervous system. Oligodendrogliocytes take part in the formation of myelin sheath in the central nervous system. So this was about the function of the macroglia, the astrocytes, Schwann cells, and oligodendrogliocytes. And this was a brief introduction of the neuroglia. Now we come to the neurons. Neurons are the building blocks in the nervous tissue. So in the nervous tissue, neurons are present. There may be 100 billion of neurons present in the nervous tissue. 100 billion neurons are present in the nervous tissue. You see the neurons consisting of the cell body, the soma, and two types of processes. So neurons is composed of the cell body and two types of the processes. Size of the cell body is variable. It contains the nucleus, you see, the nucleus, other cell organelles, the mitochondria, endoplasmic reticulum, Golgi apparatus, and other important organelle in the neuron cell body are the nissal bodies. So nissal bodies or nissal granules are present in the cell body. What are these nissal granules? Nissal granules or nissal bodies are chromophil. These are stained by the basic dyes, and these are composed of ribonucleic acid, RNA, and proteins. So nissal bodies are composed of the RNAs and proteins. What is their function? So nissal bodies, nissal granules, they synthesize proteins. Size and number of nissal bodies depend upon the physiological condition of the neuron. So size and number of nissal bodies depend upon the physiological condition of the neurons. Fatigue, exposure to toxic substance, nerve fiber injury, nerve fiber section, it results into breakdown of nissal bodies. So I told you, fatigue, exposure to toxic substance, nerve fiber injury or section, it causes the nissal bodies to break up and disappear. And this is called chromatolysis. Chromatolysis is the breakdown and disappearance of these nissal bodies when there is fatigue, exposure to toxic substance, or there may be nerve fiber damage or nerve injury or nerve section. Right? So this is about the cell body. I told you that in the neuron there are two types of processes. The dendrites and the axon. So dendrites and axons are the two types of the processes. Dendrites may be one or multiple, more than one. These are branching. So dendrites may be one or more than one, and these are branching. The other one is the axon, a nerve fiber. Axon, a nerve fiber. You see the axon, a nerve fiber. So axon is always one. In a neuron, there is always one nerve fiber. So one nerve fiber only in a neuron. And this is the axon or nerve fiber. The part of the cell body from which axon arises is called axon hillock. You see, this is the axon hillock, elevated part of the cell body from which axon arises. This is the axon hillock. Axon, the nerve fiber, which is one in a neuron, has a membrane that is exodermal, and then the cytoplasm is called exoplasm. In the exoplasm, missile bodies are not present. Mitochondria are there in the exoplasm, but no missile bodies, no missile granules present in the exoplasm. Now at the end, the axon, it divides into terminal branches having knob-like ends. So at the end, the axon nerve fiber, it divides into terminal branches having knob-like ends. And these are called synaptic knobs, or you say axon terminals, or end foot synaptic knob, or maybe simply the nerve fiber terminals. Right? I told you that axon is the nerve fiber. Right? And the nerve fiber may be myelinated or unmyelinated. So the nerve fiber may be myelinated or unmyelinated. When you say nerve fiber is myelinated, it means the nerve fiber is covered by the myelin sheath. So when myelin sheath covers the nerve fiber, it is myelinated. And when nerve fiber is not covered by the myelin sheath, it is unmyelinated. It is unmyelinated. What is myelin sheath? Myelin sheath consists of layers of Schwann cell membrane, compressed concentrically around the nerve fiber. So myelin sheath consists of layers of the Schwann cell membranes compressed concentrically around the nerve fiber to form the myelin sheath. You see the nerve fiber. This is the Schwann cell surrounding the nerve fiber. This Schwann cell will grow, will rotate around the nerve fiber, and will lay down its cell membrane. And these cell membranes are wrapped around many times. And then these cell membranes of Schwann cells, these are compressed concentrically to form the myelin sheath. You know, myelin sheath is composed of lipids and proteins as the composition of the cell membrane, composed of the lipids and proteins. Lipid may be phospholipids, cholesterol. The outermost layer of the myelin sheath, the outermost layer of the myelin sheath is the outermost cell membrane of the Schwann cell. So outermost layer of the myelin sheath is the outermost Schwann cell membrane. This is the axon nerve fiber, and this is the myelin sheath, and this is the outermost layer, outermost cell membrane of Schwann cell. This is called neurolemma. So the outermost layer of the myelin sheath is called neurolemma. Under neurolemma, we see the Schwann cell nucleus is there under the neurolemma. So neurolemma is the outermost layer of the Schwann cell membrane in the myelin sheath, and under the neurolemma we see there is the nucleus of the Schwann cells. I told you when the nerve fiber is covered by the myelin sheath, it is myelinated. It is myelinated. Keep in mind that myelin sheath is not continuous throughout the length of the nerve fiber. Myelin sheath is not continuous throughout the length of the nerve fiber. It is interrupted, it is absent at intervals. You see? It is absent at intervals, and the part of the nerve fiber where myelin sheath is absent is called node of Ranvier. Node of Ranvier. So node of Ranvier is the part of the nerve fiber where myelin sheath is absent. But at node of Ranvier, neurolemma is present. At node of Ranvier, neurolemma is present. What is neurolemma? The outermost layer of the cell membrane, or you say it is simply the cell membrane of the Schwann cell. So at node of Ranvier, neurolemma is present. Keep in mind, myelin sheath has an insulating property. Has got an insulating property. Myelin sheath is not permeable to ions. Myelin sheath is not permeable to ions. Current can't flow through myelin sheath. The current can't flow through myelin sheath. But at node of Ranvier, where myelin sheath is absent and only neurolemma is present, keep in mind, neurolemma is permeable to ions. Neurolemma has got ion channels. So at node of Ranvier, the membrane, the neurolemma is permeable to ions. Ions can move across at the node of Ranvier. But ions can't move through the myelin sheath. But these can move through the node of Ranvier where there is no myelin sheath, but the neurolemma is present there. The portion of the nerve fiber between two nodes of Ranvier is the internode. Right? So node of Ranvier, these two are nodes of Ranvier, in between is the internode. The length of the internode, it varies, say between 0.1 to 10 millimeter. 0.1 to 10 millimeter is the length of the internode. Or you say the distance between two successive nodes of embryo varies between 0.1 to 10 millimeter. 0.1 to 10 millimeter. Keep in mind that thicker the nerve fiber, longer will be the internode. Longer will be the internode, and faster will be the velocity of conduction of nerve impulse. I am repeating, thicker the nerve fiber, longer will be the internode, and faster will be the velocity of conduction to the nerve fiber. So thicker the nerve fiber, longer will be the internode, so faster will be the velocity of conduction to the along the nerve fiber. So don't forget, neuroderma is the outermost cell membrane of the Schwann cells. Neuroderma is present at the node of embryo, and nodes are highly permeable to ions. But myelin sheath is not permeable to ions. Now, surrounding the neuroderma, there is a connective tissue covering which is called endoneurium. Surrounding the neuroderma, there is a connective tissue covering which is called endoneurium. So endoneurium is connective tissue covering surrounding the nerve fiber around the outer to the myelin sheath. Bundles of nerve fibers are covered by a layer of connective tissue called perineurium. So perineurium is the connective tissue covering around the bundle of nerve fibers. So around the bundle of nerve fiber, there is a connective tissue covering which is called perineurium. And then the nerve is covered by a covering of connective tissue which is called the epineurium. Epineurium is the connective tissue covering around the whole nerve. So surrounding the nerve fiber, endoneurium. Surrounding the bundles of nerve fibers, perineurium. And surrounding the whole nerve, the covering is called epineurium. So this was about the myelin sheath and the two types of nerve fiber. Keep in mind that nerve fibers which are unmyelinated, these are surrounded by Schwann cells, which have not wrapped the myelin sheath. So nerve fibers which are unmyelinated, these are just covered by the Schwann cells which have not wrapped around the myelin sheath, around nerve fiber, and these nerve fibers are unmyelinated. These are unmyelinated. Now I've discussed about the formation of myelin sheath, and it's called myelinogenesis. So formation of myelin sheath, that is called myelinogenesis. In the peripheral nerves, myelin sheath is formed by the Schwann cells. Schwann cells, these form the myelin sheath in the peripheral nerves. You see, this is the Schwann cell. This is the axon and nerve fiber. The Schwann cell surround the axon and nerve fiber. And then the Schwann cell which surround the nerve fiber, it starts growing, it go on rotating around the nerve fiber, it goes on rotating around the nerve fiber and lay down its cell membrane. Lay down its cell membrane many times. And these cell membranes of the Schwann cells become compressed concentrically to form the myelin sheath. So these layers of the cell membrane of Schwann cells are compressed concentrically to form the myelin sheath around the nerve fiber. You see, this is the Schwann cell. It is growing, rotating around the nerve fiber and laying down its cell membrane many times. So these cell membrane layers, these become compressed concentrically around the nerve fiber to form the myelin sheath. And keep in mind, the outermost cell membrane, outermost layer of cell membrane of Schwann cell, this is called the neurolemma. This is the neurolemma. Under neurolemma, you will see the nucleus of the Schwann cell. So under neurolemma, you can see the nucleus of the Schwann cell. So this was about the formation of myelin sheath. I told you that Schwann cells, these form myelin sheath in the peripheral nerves. And keep in mind, Schwann cells retain the ability to form myelin sheath throughout life. Schwann cells retain the ability to form myelin sheath throughout life. Schwann cells retain their ability to form myelin sheath throughout life. So after birth, formation of myelin sheath in the peripheral nerves is completed in 2 years after birth. So in 2 years after birth, formation of myelin sheath around the nerve fibers in the peripheral nerves is complete. And keep in mind, Schwann cells retain their ability to form myelin sheath throughout life. In the central nervous system, myelin sheath is formed by the oligodendrogliocytes. So oligodendrogliocytes, these form the myelin sheath in the central nervous system. You see, the oligodendrogliocyte, right? It forms myelin sheath or nerve fibers in the brain, spinal cord, that is central nervous system. So oligodendrogliocytes, these form the myelin sheath in central nervous system. I told you about Schwann cells forming the myelin sheath in peripheral nerves. Schwann cells, one Schwann cell form myelin sheath in one internode. So in one internode, myelin sheath is formed by the Schwann cell. So Schwann cells form myelin sheath in one internode. But in central nervous system, oligodendrogliocytes. In central nervous system, oligodendrogliocytes take part in the formation of myelin sheath around a number of nerve fibers. In central nervous system, oligodendrogliocytes, they take part in the formation of myelin sheath around a number of nerve fibers. While in the peripheral nervous system, in the peripheral nerves, one Schwann cell, it forms myelin sheath in one internode. But of course, in the central nervous system, one oligodendrogliocyte, it takes part in the formation of myelin sheath along the number of nerve fibers. Oligodendrogliocytes. Once these form myelin sheath in the early life, lose their ability to form myelin sheath again. So oligodendrogliocytes, once these are formed the myelin sheath in the early life, lose their ability to form the myelin afterwards. So that is why regeneration is possible in the peripheral nervous system, but not in the central nervous system. So keep in mind, the regeneration is possible in the peripheral nervous system, but not in the central nervous system. Why? Schwann cells retain their ability to form myelin sheath in the peripheral nervous system. Oligodendrogliocytes, once these have formed the myelin sheath in the early life, lose their ability to form the myelin sheath again. So this is the reason why in the central nervous system there is no regeneration, but it is possible in the peripheral nervous system. I have discussed about the nerve fibers, myelinated and unmyelinated. Those covered by the myelin sheath and those which are not covered by the myelin sheath. Which nerve fibers are myelinated, which are unmyelinated? I give you a basic hint to remember. All the autonomic preganglionic nerve fibers, all the autonomic preganglionic nerve fibers and somatic nerve fibers having diameter more than 1 micrometer and somatic nerve fibers having diameter more than 1 micrometer are myelinated. So which nerve fibers are myelinated? All the autonomic preganglionic nerve fibers and somatic nerve fibers having diameter more than 1 micron or micrometer. Now which nerve fibers are unmyelinated? Which nerve fibers are unmyelinated? These are autonomic nerve fibers which are post-ganglionic. Autonomic nerve fibers which are post-ganglionic and somatic nerve fibers having diameter less than 1 micrometer. So which nerve fibers are unmyelinated? Autonomic nerve fibers which are post-ganglionic and somatic nerve fibers having diameter less than 1 micrometer. These are unmyelinated. I told you about the nerve fiber. Nerve fiber is the axon. An axon or nerve fiber in a neuron is always one. Always one. Length of the nerve fiber is variable. It may be a few micrometer, micrometer. It may be up to 1 meter length. Its length may be up to 1 meter. So length of the axon is variable. It varies between few micrometer to 1 meter. Say the sciatic nerve fiber takes origin from the lumbosacral spine, and then these go down to the foot. So the length will be maybe up to 1 meter. 1 meter. In the brain, some of the neurons have got axon nerve fiber having a length of the few millimeter, for a few micron, right? So length is variable. It varies between few micrometer to 1 meter. So this was about simple. Now keep in mind one thing, that in the cell body of the neuron, there is no centriole or centrosome. So no centriole or centrosome in the neuron. So neurons can't regenerate. Neurons can't regenerate. Why? These don't contain centriole or centrosome. So these don't contain the centriole or centrosome, so these can't regenerate. Now I discuss about exoplasmic transport or axonal transport. Exoplasmic transport or axonal transport. So exoplasmic transport or axonal transport. Keep in mind that anatomical and functional integrity of the neuron, of the nerve fiber, it depends upon the cell body. So anatomical and functional integrity of the nerve fiber, it depends upon the cell body. I repeat that anatomical and functional integrity of the axon, it depends upon the cell body. Vesicles, certain proteins are synthesized in the cell body. Vesicles and certain proteins are synthesized in the cell body. And then these are transported in the exoplasm to the nerve terminal. These are transported in the exoplasm to the nerve terminal. And this is called the exoplasmic transport or axonal transport. It is exoplasmic transport or axonal transport. So this transport of the vesicles, proteins, other materials which occurs in the exoplasm, this transport occurs along the microtubules. You know there are microtubules in this cell which form the cytoskeleton. So these microtubules, these provide the track. These provide the track for the transport of substances in the exoplasm. So the microtubules, these provide the track for the transport in the exoplasm. Now in addition, there are certain motor proteins, certain motors, which help in this transport. So in the transport, certain protein motors, these help. And these include the kinesin and dynein. So kinesin and dynein are the protein motors which help in the transport of substances, transport of vesicles, transport of protein, other substances in the exoplasm. So these are the protein motors, the kinesin, you see, protein, and similarly the tiny proteins are the, and this is the microtubule which help in the transport in the exoplasm. So kinesin and dynein are the protein motors which help in the axonal transport or exoplasmic transport. Now this axonal transport is of two types. So two types of axonal transport: orthograde or anterograde. Orthograde or anterograde. Orthograde or anterograde. So this anterograde or orthograde transport, it occurs from the cell body. That exoplasmic transport is of two types: anterograde or orthograde, and second type is the retrograde. Anterograde or orthodromic transport, it occurs from the cell body towards the nerve terminal. So orthograde or anterograde transport, it occurs from the cell body to the nerve terminal. This is the anterograde or orthograde transport. Orthograde transport. The other transport is retrograde. That is transport in the reverse direction, and that is from the axon terminal toward the cell body. From the axon terminal toward the cell body. I told you the example of the anterograde or orthograde transport, and that is from the cell body to the axon terminal. So this retrograde transport, it occurs from the axon terminal to the cell body, and by the retrograde transport, recycled vesicles, viruses, and nerve growth factor are transported by the retrograde transport. Recycled vesicles, nerve growth factor, and viruses are transported by the retrograde transport. The velocity of this axonal transport, it varies. In this transport, there are two types of the transport systems: slow transport and fast transport. So in the exoplasmic transport, there are two type systems: slow transport and the fast transport. Slow transport can transport at the rate of 0.5 to 10 millimeter per day. So in the slow component of the transport, the slow component of the exoplasmic transport, the velocity is, the rate is 0.5 to 10 millimeter per day. In the fast transport, exoplasmic transport, the rate of velocity is 400 millimeter per day. So it is 400 millimeter per day in the fast transport system in the exoplasmic transport. So I told you that in this exoplasmic transport, this anterograde or orthograde transport, that is from the cell body to the axon terminal. And the vesicles and the proteins, some chemicals are transported in this system, that is anterograde. In the retrograde transport, the recycled vesicles, viruses, nerve growth factor are transported from the axon terminal to the cell body. And then this system of transport in the exoplasm has got the slow component which can transport at rate of 0.5 to 10 millimeter per day, and fast system which can transport at rate of 400 millimeter per day. So this was for today's, right? So discuss tomorrow inshallah next time.